It's a great pleasure to be here this month again with all of you, but also in particular with Lars, which is an esteemed member of the Legume Society. Lars is an associate professor at Curtin University. Is also very kind because right now is nighttime at his place, but he still kept on and decided to be our guest. And today we are going to talk about Ascochyta blight in lentil. So, Lars, thanks for being here, and please take the mic.
Thanks, Nelson. Thanks for the very kind introduction, and it's a great pleasure to be telling a little bit about our work that we do in the Center for Crop and Disease Management that's focused around Ascochyta blight in lentil. So actually get the slides to work. There we go. So what I thought I'd do first is tell you a little bit about our center. So it's essentially a co-investment between our university, Curtin University, and one of the main funding bodies in Australia, in the Grains Research and Development Corporation. And the main aim of our centre is to reduce the economic impact of the diseases that our grain growers face. And we do that in 3 different themes of research. So the first one focuses on fungicide resistance and trying to identify and characterize mutations that lead to fungicide resistance as they occur in crops in certain pathogens. The second theme focuses on cereal diseases. That's mainly wheat and barley, and we started some work on oath pathogens as well. And then the theme that I lead focuses on canola and pulse diseases, so broadleaf crops. And we do this in a dedicated research facility that you can see here in the image. We've got 3 floors of laboratory space with built-in plant growth facilities as well. And then the university has a field trial area with glass houses and controlled environment rooms and a netted area as well. So in terms of pulse diseases, our main disease that we focus on in the center is Ascochyta blight, and we do research on Ascochyta blight in both chickpeas and lentils. We're also doing some work on Sclerotinia stem rot, predominantly in canola, but more recently also in narrowleaf lupin, which is one of the key grain legumes in rotation in Western Australia with wheat. And in July of this year, we also started working on Botrytis grey mold in chickpeas and lentils. So they're the key crops and pathogens that we work with in terms of legumes. But today I'm just going to focus on our Ascochyta blight research in lentils. So with the Ascochyta species, they're all host specific. So an Ascochyta of lentil can only cause disease on lentil and not on any of the other legumes. And it's found in all lentil production areas around the world. And in the case of Australia, it's our most costly disease, followed by Botrytis grey mold. And so that's why we're trying to identify sources of resistance and try and work with breeders to bring those resistance sources into the varieties that they're breeding, and obviously try and understand the mechanisms of pathogenesis. By and large, Ascochyta blight in lentils is well controlled by fungicides, but also cultural practices. But obviously, the preferred mechanisms to try and control that be through genetic resistance in the varieties. So over the years in Australia, certain resistance sources have been overcome and are no longer as effective as they used to be, and there's a clear pathotype structure. And that's what our research has been focusing on. So it's a high value crop in Australia, lentils, and we mainly export it to the Asian continent. And it's known, Australian lentils, for its high protein content. But as I mentioned, Ascochyta lentis is the main disease in Australia that's causing disease issues and can cause up to 50% yield loss if a crop is not protected and it's a susceptible variety. So quite a serious disease if left untreated. And so our approach focuses on both the host, lentil, to try and identify sources of resistance, but also the pathogen Ascochyta lentis and the interaction. So, and I'll talk about that in the coming slides. And so I'm going to introduce some key players in our research, mainly focusing on two pathogens or pathotypes. The first one is an isolate called ALQ, and the other one is an isolate called P9424. What's interesting about these guys is that they have different responses, as you can see in these images here, on different host genotypes of lentil. So ALQ doesn't show any disease on nipper, but is pathogenic on PBA hurricane, and vice versa. P9424 is pathogenic on nipper, but doesn't cause disease on hurricane. So if we put that in some schematics, unhappy interactions are the nipper by P94 and the cure by hurricane interactions, or in other words, they are compatible interactions where the pathogen is able to cause disease, whereas the others are incompatible interactions that lead to resistance. And so that makes it a tractable system for us to try and just dissect the key genes in the pathogen, but also the key genes on the host side of the interaction. And so that's been a key focus in our team. So most research groups tend to make crosses between resistant and susceptible plants and then dissect the genetic resistance. We did that, but we also did that on the pathogen side. So in collaboration with colleagues at ILSW-s3e3 in South Australia, we've developed a biparental fungal mapping population derived from those two parents, P9424 and ALQ. And what you can see here in this graph is the disease score on the y-axis, and then the offspring of that cross, as well as the two parents in the darker bars with their responses on hurricane in green and nipper in yellow. So we phenotyped about 100 of these progenies on both parents and then conducted QTL analysis. And that's what you can see in this graph here. So we've got the lot score on the y-axis, and on the x-axis, the 23 chromosomes of Ascochyta lentis. And what you can see quite evidently is that there's a very strong locus on the tail end of chromosome 3. So we wanted to see what kind of genes in the pathogen underlie that locus. And what we found when we zoom into that region is that there is one particular gene here that's in a typical AT-rich region with lots of retrotransposons and transposable elements. And that gene was different between the two parents, QL and P9424, as illustrated in this green bar where you see the dips. There is differences in the sequence alignment. So we wanted to look at that particular gene in a bit more detail and see if these variances in the sequence correlated with the disease. And what we found was that if you are pathogenic on hurricane, you carry the upper band. In all cases, if you see green disease, you have that upper band. And that was a perfect correlation with that phenotype. So what we decided to do is to clone that gene and put that into agrobacterium and then agro-infiltrate lentil leaves. And we did that with both versions. So the version from QL and from P9424. And then we had a necrosis-inducing control protein from the pathogen called NLP2. And a negative control in green fluorescent protein. What you can see here on nipper is that NLP causes necrosis, but neither of the versions of the protein cause the necrosis response in nipper. Whereas in hurricane and bolt, which are two varieties that carry the same resistance source, you can see that there was clear necrosis when we infiltrated with ALAVR1-1, but not with ALAVR1-2. The same held true for both. And so the ALAVR1 protein, 1-1 protein, was acting as an avirulence effector that triggers resistance in PBA hurricane XT, or at least that was our working hypothesis. So to prove that, we can manipulate the pathogen and knock genes out. And we did that in both pathogens. So Cure, which is not pathogenic on Nipah, as you can see here, when we knock out the ALAVR1 protein, nothing changes. But what was interesting with the Cure isolate, when we knocked out that ALAVR1 version, we see a reduction in pathogenicity on PBA hurricane XT, or in other words, that version of the protein was acting as a potential virulence factor. In contrast, when we knock out the gene in P9424, we did not see any significant difference relative to the wild-type phenotype on Nipah, which is pathogenic. But interestingly, when we knock out the gene and inoculate the knockout on PBA hurricane, which the wild-type version recognizes the protein that leads to resistance, you can see that all of a sudden the pathogen has the ability to cause disease. In other words, it acts as a true avirulence gene. So this was done in a classical biparental mapping population, but what we did side by side is annual collection of Ascochyta lentis isolates from around Australia. And so we've been doing that since 2013. And so what we did with a collection of isolates collected between 2013 to 2018 is inoculate about 180 of these isolates on a set of differential lentil genotypes, which are listed here. And what you can see here again is that for both hurricane, Indian het, and Nipah, there's a strong association with that locus on chromosome 3 that we previously identified to carry the ALAVR1 resistance gene. Now that avirulence gene interacts with the corresponding resistance gene in hurricane, but there's a second effector that interacts with the resistance gene in Nipah. So whilst we didn't see any other loci elsewhere when we used the SNP markers, when we used the silico dart markers, there was a presence-absence variance on one of the other chromosomes as well that might be of interest, and we're currently investigating the candidate gene that underlies this region, as well as the neighboring genes of ALAVR1. But when we took the ALAVR1 protein gene and our markers that can distinguish the two forms, so 1-1 and 1-2, we previously showed you that that holds up nicely with hurricane pathogenicity. We did the same on that isolate collection, and again, what we found was that if you were pathogenic on hurricane, you had that upper band, and that's what you can see. And when we start looking at a historical shift of genotypes of lentil that have been introduced since about 2015, we see a shift from pathotype 1 isolates that are pathogenic on genotypes like Nipah, and a shift to pathotype 2 isolates that have been able to overcome that resistance source that you see in PBA hurricane and varieties like Bolt. So we were able to develop this PCR-based approach as a diagnostic marker to distinguish the two pathotypes, so pathotype 1 and pathotype 2. The question we then had, well, that's great. We know what it looks like at a national context in Australia, but how does that look at a global level? And so we started collaborating with a range of colleagues from around the globe to try and obtain isolates from different lentil producing countries. So with colleagues from Agriculture and Agri-Food Canada, so Lona Bookwell's team, we got isolates from Washington State University, from Wei Dong Chan's lab, from colleagues in Akata, so Saeed Kamal and his fellow pathology team, and from Canan Chen at Gaziantep University in Turkey. And so we're starting to develop an international collection of lentil isolates. So if you have lentil isolates in your collection that are not on this list, we would love to hear from you and obtain those isolates. Because one of the other activities that we're currently working on in our lab is to sequence these and sequence a diverse panel of Ascochyta lentis isolates from around the globe. So these are the ones that we've sequenced to date, about 151 of them, but we'd love to get a better representation of some of the other countries, particularly in Europe and in South America and Africa, where we're still a bit thin on the ground, and in certain parts of Asia as well. So when we looked at the ALAVR1 lockers, and started pathotyping those for the 2 different pathotypes with our primers, we were able to identify that in most of the lentil producing countries where we had a decent sample size, both pathotypes are present. So in Canada, US, Turkey, and Syria, where a lot of lentils are produced, and obviously in Australia, we see both pathotypes. But as you can see, in some of the samples that we were looking at, some of our primers weren't working. And we're wondering why that was. Is the gene absent, or are there variants in the ALAVR1 gene? And so we used full-length sequencing of the genes with Sanger sequencing to sequence the different genes across the international isolates, and that's depicted in this phylogram here. And so you can see that there's quite a few different versions of the pathogen of the ALAVR1 lockers that we've identified, and so that raised the question: which versions are being recognized by the resistance gene in varieties like PBA hurricane and bald, and which ones are not? And so we cloned those different versions of the ALAVR1 protein into agrobacterium and did agroinfiltrations again. And what we found was that, again, ALAVR1-1 held up. It produced strong necrosis in both PBA bald and PBA hurricane. And then there was an isolate, a set of isolates from Canada that had a single base pair change that led to an amino acid change that also caused necrosis in both varieties. Then there was a variant in Syrian isolates that also caused necrosis, but a lot weaker than what you see for these 2 versions of the protein. And then interestingly, there was a quite distantly related Ascochyta pathogen of vetch called Ascochyta viciophilosa that had a homolog with about 81% identity that also caused necrosis in both PBA bald and to a lesser extent in PBA hurricane. So that was quite interesting. We also found another variant in an isolate from Morocco that was more similar to the 1-2 that acts as a virulence factor that indeed didn't cause necrosis on either PBA bald or hurricane. So now that we've got an understanding of the different forms of ALAVR1 and which versions of the protein are being able to be recognized by the resistance gene and which versions are not, that allows us to develop a diagnostic test for quick pathotyping of Ascochyta lentis isolates. So traditionally, that kind of research takes us a long time. We get an infected sample, we surface sterilize that, we plate that out, and then single spore that, and then of the single spored isolates, we do a DNA extraction, high quality DNA extraction, and then the endpoint PCR. That's quite a time consuming exercise to get that. So we wanted to see if we could speed up that process. So obviously the single sporing is quite time consuming. So if we take that step out and just take a sample from the field, once it's arrived, we can do a good quality DNA extraction, and then try and see if we can do a quantitative PCR, because that then will allow us to identify the ratios of pathotypes 1 and 2 of a sample that might be sent in by a grower. So, but ideally, where we want to get to is a super quick method whereby we take those samples, we grind it in a quick DNA extraction method that takes about half an hour, and then straight away go to quantitative PCR. So to try and do that, we had to develop an assay that was specific, it was sensitive, and diagnostic. And so the way we did this is we designed primers that are conserved across all the variants of the ALAVR1 gene. And then we designed different fluorophores that are diagnostic between those that are recognized and those that are not recognized by the resistance source in PBO, hurricane, and bolt. And that's what we see here when we look at the analytics. We can distinguish pathotype 1 from pathotype 2, and it's quite sensitive. So we can go down to picograms of DNA of the pathogen, and we can still recognize and detect it. So that was great. The other thing that was great was it can distinguish the different versions and the different isolates from overseas that have the variants that we don't have present in Australia as well. So this methodology can be adopted in overseas diagnostic labs if they wish to do so as well. The other thing that we wanted to make sure was that we're not capturing any off targets. And so we use those specific primers here to try and see if it amplifies in any other host pathogens of pulses. As you can see here, that's not the case. And then we looked at other Ascochyta species, and the only other Ascochyta species that it can amplify in is that Ascochyta pathogen of vetch that I mentioned earlier. And again, here you can see that it can amplify the different variants of the international isolates that we've identified today. So that's great. So that's working well, and we can now receive samples for growers and advise them of the ratios of pathotype 1 to 2 that they might have in their paddock, and make more informed decisions around what varieties they may wish to plant based on the pathotype dynamics and population in their paddocks. But as I mentioned, there was a homologue in a related pathogen. And so an honest student in our team generated some whole genome sequence data sets using Oxford Nanopore. And what she found when she was working with isolates of vetch and fava bean was that this Ascochyta vitiofilose pathogen, its genome is more closely related to Ascochyta lentis than it is of a related Ascochyta species called Ascochyta vitia, which can also cause disease on vetch. The latter is more closely related to Ascochyta vaba. So when we started to look at the arrangement of the ALAVR1 homologue in the vetch-infecting pathogen called Ascochyta vitiofilose, you can see in blue here the regions that are highly synthetic and the gene content that's conserved between the two versions of Ascochyta lentis and this Ascochyta vitia. You can see that on both sides there is good synteny and that the gene is present. But in this Ascochyta vitia, you can see that there's conservation on the left-hand side of that ALAVR1 region, and then on the right-hand side, which is the tail end of the chromosome, remember, the end of chromosome 3 in all these pathogens, that homology and that synteny is lost. So we wanted to know what that version in Ascochyta vitiofilose was doing in vetch. And so what we've got here is some disease assays that the student conducted on 4 genotypes of common vetch and 3 genotypes of hairy vetch. She assessed these with disease scores, with macroscopic phenotyping of the lesions that were developed. But what you also can see here is the microscopy, which quite elegantly shows the differences. So on common vetch, Ascochyta vicious is unable to cause disease. In fact, when the pathogen is trying to penetrate the host cells, it causes a hypersensitive response and it restricts the pathogen from entering the host plants. Whereas on hairy vetch, Ascochyta vicious can cause disease, as illustrated here by the spreading necrosis that you see here. This is after 7 days, and that still holds true after 14 days. Now, the related pathogen called Ascochyta vicious is able to cause disease at both 7 and 14 days. And then below here, we've got a tween control where you can see no necrosis and no pathogen present. So our hypothesis then was, could the AVV-AVR1 gene in Ascochyta vicious lead to this hypersensitive response that we see in common vetch and restricts its host range to just common vetch and not just hairy vetch. So what we did is we knocked out the AVV-AVR1 gene in Ascochyta vicious. So here we got the wild type. Again, you get those nice hypersensitive response at the sites where it's trying to penetrate, but the knockout is completely pathogenic now on common vetch and on all 4 genotypes of common vetch that we've tested. When we then put the gene back in the complementation strain, you can see that that hypersensitive response is restored. And so the AVV-AVR1 gene mediates host specialization of Ascochyta vicious. So that was a nice finding. So what are some of the other things that we're trying to do with these virulence proteins? But one of the things with the agro-infiltration methods is it allows us to trace the parentage of lentil genotypes in the breeding program and trying to identify where that resistant source is coming from. And what you can see here is that PBA bolt that I mentioned in Hurricane, they cause that hypersensitive response, and we're now down to a bunch of parents in black here that just have the writing on it that we've got to find out whether they have that hypersensitive response. We've obtained these lines now, and we're trying to get that material ready for agro-infiltration. The other thing that we want to try and do is actually generate purified protein. Agro-infiltration of lentils is quite finicky, and you got to do it at the right developmental age. Otherwise, you really struggle to get good phenotypes. So that's something that we're working towards at the moment in the lab. That's just one effector and one gene. I mentioned before that we're trying to identify additional effectors through whole genome sequencing and generation of a pangenome. But one of the early things that we did is compare the 2 genomes of our 2 favorite Ascochyta lentis isolates in ALQL and P9424. And what you see here in this circus plot is the 23 chromosomes of the pathogen. And what you can see is that essentially the chromosomes are highly comparable, and there's very little rearrangements or translocations. What we've got here on the right-hand side of the graph is again the 23 chromosomes and the mitochondrial genomes that we've been able to assemble. And what you can see with the black dots here on the end is the telomeric ends. And so you can see that with the Oxford nanopore sequencing that we've done, we can pretty much assemble whole genomes telomere to telomere bar 1 chromosome end, and that's the end where there's a lot of the ribosomal DNA. The other things that we've depicted here in blue is the gene content, and then in purple, candidate other genes that might be involved in pathogenicity that we call effectors. You can see that some of these are present in both, but then in other instances, they are unique to one particular pathogen species or isolate. And so that's quite interesting. The other thing that I would like to point out here is that you see these little red dots here, and they're a type of transposable element that sit on the end of the chromosome called PLEs. And we found those only in this P9424 isolate and not in ALQL. So that's another interesting finding that we're doing some more work on. So yeah, the way these PLE transposons work is they attach to the end of chromosomes, and therefore rearranging what's happening on those tail ends. So we ran our genome annotations through what we call an effector prediction pipeline to try and identify other genes that might be important for pathogenicity or causing disease. And so a quick way to screen these candidate effectors for their ability to cause necrosis or a hypersensitive response is to agroinfiltrate them in tobacco, which is a lot more readily able to conduct these transient assays in. What you can see here is, again, how we do these assays. So we've got our green fluorescent protein when we put the right filters on, fluorescing green. And then we've got, in this case, 3 versions of NLP, the Ascochyta lentis one, the Pyranella panotis one, and the Ascochyta rabii that can cause necrosis when we look at it in the bright field. And so we took our top 12 candidates, and what you can see here in these graphs is one where we've infiltrated the protein with its signal peptide and without its signal peptide to try and see how important it is to have the signal peptide for its ability to cause necrosis. But the take-home message essentially is all but, candidate number 10 were able to cause necrosis in Nicotiana benthamiana. In other words, these are not host-specific necrosis-inducing proteins. So, to summarize the first part of my talk, we've characterized a number of effectors, and ALAVL1 is an avirulence gene that triggers an immune response in those lentil cultivars that carry the resistance gene that can recognize this ALAVL1-1 protein. We've demonstrated that ALAVL1-2 contributes to virulence in Hurricane XT. We also used GWAS analysis to confirm that this association occurs and that there's likely other resistance interactions with other lentil genotypes co-located in that chromosome 3 region of the pathogen. So that seems to be a hotspot for potential effectors. I showed you that there's 6 forms of this ALAVL1 protein, but only the clade that contains the 1-1 protein elicits a hypersensitive response in cultivars that carry the resistance gene, and we've been able to develop a high throughput laboratory test to distinguish and quantify the 2 pathotypes. This agroinfiltration and purified proteins that can be used to try and trace the receptor in lentil breeding programs, and this is something that we're actively working towards developing in the lab. And lastly, we're working towards the generation of an international Ascochyta lentis pangenome. We'd love to receive isolates from colleagues on the call if they have them. So that was the host site. But what about the pathogen side, but what about the host side of the interaction? So we're focused on 4 different resistance sources. So obviously our favorite 2 in Nipah, indicated in green there, PBA bald, which is the same as hurricane. The purple lines indicated here all carry that same resistance source. Indian hat, which is a different resistance source again, and then ILL-7537 in this corner here. And then there is ILL-6002, which is highly susceptible to both pathotype 1 and 2 of Ascochyta lentis. And so we employed 3 populations to try and evaluate these populations with both pathotypes, the 6002 by Indian hat, the Nipah bald population, and 6002 by ILL-7537 population. So I'll go through each of these populations and discuss the phenotyping results. So the way we've done this work is we make the crosses, then generated recombinant inbred lines, We sampled the tissue at the F5 stage and then collected the F6 seeds that we phenotyped, and the tissue from the F5s we genotyped on the Pole SNP chip that was developed by colleagues at Agriculture Victoria. That allowed us to then do the linkage mapping and QTL analysis. And so one of the things that we wanted to know was how well our genetic maps would correlate with the physical maps, and that's pretty much what we see here. And it's by and large that it correlates quite well, but there are some rearrangements and inversions that do occur on some of the chromosomes. So once we finished phenotyping these, we do this typically in seedling assays because they're quick and high throughput for us. We grow the seedlings up for a few days, then we inoculate them, and then we score them after 7 to 10 days. And so when we do that for these populations, we can inoculate them with both pathotypes and conduct a QTL analysis. And in this case, we see a strong locus with a lot score of greater than 30 on the short arm of chromosome 2 for pathotype 1. And we see 2 other minor loci on chromosomes 3 and 5. And for pathotype 2, we see 2 minor loci on 2 and 7. Whilst we were bulking up this population in one of our hoop houses, we also had a powdery mildew outbreak, and we scored the population for mildew as well and identified a strong locus on the short arm of chromosome 3 for powdery mildew resistance. So that was a nice little bonus. So this was the genetic map that I showed you here, and what I'm showing you here is the actual physical map when we placed the markers in the order on the physical map, and essentially nothing changed. What's interesting is when we do that, it allows us to compare it to other maps and other populations that have previously been phenotyped, and the loci here indicated with the stars were identified by other research teams previously, whereas the others are obviously nothing. So the second population that we then inoculated was the nipple ball population, and again we found a very strong locus for pathotype 1 on chromosome 2. The third population that we looked at was ILL-75-37. We're particularly interested in this population because it's got strong resistance to both pathotypes 1 and 2. And this was a population that was developed in collaboration with the University of Melbourne, Griffith University, and University of Western Australia. And this population we inoculated with 4 different isolates. 3 pathotype 2 isolates and 1 pathotype 1 isolate. And again, what we found was a really strong locus on the short arm of chromosome 2 that was effective against both pathotypes 2 and pathotype 1, as you can see here. So these seedling assays are great. They give us a quick answer. It's high throughput, but how does that correlate with the field? Well, we evaluated the same population in the field with both pathotype 1 in orange and blue pathotype 2. And again, we find the main locus on chromosome 2 for both pathotypes 1 and 2. And we found a secondary locus on the short arm of chromosome 3 for pathotype 2 as well. So what we find in our seedling assays correlates with what we observe in the field. So for those of you paying attention, you would have noticed that in each of these populations, we identified QTLs on the short arm of chromosome 2. So that raises the question, is this just one of the same resistance source, or is this an area in the lentil genome where an original resistance gene has duplicated and diverged? So could these be the same element, yes or no? Well, we believe that's not the case, and that the latter hypothesis about an R gene cluster potentially duplicating and diverging is what's happening here. And that's mainly because we've got different pathotype responses, right? So we've got the ILL-75-37 that's resistant to both pathotypes 1 and 2. that have resistant loci there, and then you've got the pathotype 1 resistance from Indian head and bolt that are there as well. They also have different statistical boundaries, as indicated here by the highlighted colors that are non-overlapping in the case of the Indian head and the 75-37 resistance, and the mode of inheritance is different, where the one in 75-37 is dominant, and for some of the others, that's not the case. So when we then start to look at the markers distribution across this chromosome, you can see that we've got an opportunity in terms of the SNP content to try and fine map these. And that's what we did for the 75-37 resistance source to start with, and we're going to be doing the same for the other populations to truly try and identify whether these loci are different. And our approach to do this is to create what we call heterogeneous inbred families. The way we do this is the same as generating a real population, but then at the F5 stage, what we do is we select those individuals in our region of interest that are still heterozygous at the QTL locus. So in the case of the 75-37 population, there were 14 F5 individuals that were still heterozygous in that region of interest. We then started multiplying that seed, and that gives you then novel mosaics of recombination within that heterozygous site, and we obtained 160 individuals. We then genotyped those 160 individuals with markers that the PhD student M in our team generated, and that allowed us to further narrow down that region of interest. And what it allowed us to do is to narrow the 160 down to 60 that had different recombination events, as illustrated by the genetic map that you can see here, that allowed further fine mapping. So in the original gene region of interest, there were still 480 genes, of which 104 were associated with plant defense and 26 with typical MBS-LRR disease resistance genes. And then we started fine mapping to that region of interest. And we did that in 2 screens. And in the first screen, we narrowed it down to a much smaller region. And with the second screen, to an even smaller region that only contains 23 genes, including 3 of those that are classical MBS-LRR genes. And so we're hoping to try and identify the causal gene now. And the way we're doing this is through adaptive nanopore sequencing of this region of interest in both of the parents, and that work is ongoing as we speak. So to summarize part 2 of the talk, we've identified 8 AB resistance QTLs in the 3 populations, and the largest of each of these QTLs, or the largest effect QTLs, were all on the short arm of chromosome 2, and that's a hotspot for Ascochyta blight resistance, where the resistance in Indian head and PBA bolt is effective against pathotype 1, and ILL-75-37 to both. And that with the fine mapping with the HIVs, we've identified or narrowed down the region of interest for the 75-37 resistance to 23 genes, including 3 classical disease resistance genes. We've also identified additional QTLs in Indian head and some QTLs for powdery mildew. So, of course, it's nice to be able to talk to you in this webinar, but I actually did very little of this work. It was done by my awesome research team. And so I'd like to thank the people that have contributed to this work. So Johannes is our one of our research technicians that's done a lot of the transformation and generation of the knockout and complementation strains. He's doing all the sequencing work in the team as well. Christy does a lot of the field and seedling assay phenotyping. Hedy is our expert in terms of the maintenance and collection of the isolates, and did a lot of the ALAVR1 characterization work. Bethany has helped out the team a lot with the generation of a knockout by knockout population that we're working on, and Bernie is the key postdoc on the Ascochyta Lentil project that oversees that work. Rob does a lot of the work on Ascochyta arabia of chickpea, but has also helped out with various aspects of the project, including the field phenotyping. Em was the PhD student that did all the QTL analysis work, and Grace did all the work characterizing and generating the knockouts in the Ascochyta visualisae pathogen in vetch. And I'm very pleased that Nim has joined our team earlier this year as a new PhD student that's going to take on some additional work around the effective characterization and trying to identify the ALAVL1 interactors in the different lentil hosts. And so with that, I would like to acknowledge the rest of the wider CCDM team, our partners at the Lentil Breeding Program at Agriculture Victoria, our colleagues at the University of Melbourne, SARDI, DPERS, who helped with the population development and isolate collections, the Lentil Breeding Program at GIA, and Bioplatforms Australia, who've done a lot of the Illumina sequencing of some of the Ascochyta isolates that we've been working with in the lab. And then our various partners overseas that have provided us with isolates towards the development of our pangenome. And last but certainly not least, I'd like to acknowledge our funding bodies. Without them, we couldn't do this research, the GRDC and of course, Curtin University. So I'll leave you with a plug for an upcoming conference in Perth. So for those of you that haven't been, I invite you to come to the 8th International Food Legume Research Conference and 5th Australian Pulse Conference that will be held in September next year. Scan the QR code if you haven't heard about it, or go to our website. We're hoping to announce a rough outline of the program towards the end of the year or early next year, when we will also have registration and abstract submission open up in the new year. So with that, I'll stop talking, and I'm happy to answer any questions you might have.
Thank you, Lars. It was a very interesting contribution from yours, and we'll also make sure to rebound and have all the information about the conference you were talking about on our website and on our social platforms. With that, I ask if the audience have some questions. Otherwise, I will ask something to break the ice. Somebody in the chat. So somebody thanking you. You mentioned about sclerotinia effort. How is it going? Do you think that you may find a similar case with sclerotinia to Ascochyta?
Long story short, no. So obviously, Ascochyta lentis access a hemibiotroph, so it's got a latent phase, biotrophic phase, before it switches to necrotrophy. We believe that with sclerotinia, it behaves more like a true necrotroph, and in all the systems that we've looked at so far, it seems to be a highly quantitative form of resistance that you find, and it's a partial resistance. So complete resistance like you see in Ascochyta lentis in lentil, you won't see for sclerotinia. So many genes in a host genome with small effects that lead to resistance. Having said that, we've identified a number of sclerotinia effectors that seem to interact with sensitivity genes, particularly in canola, in the host. So if we can remove those sensitivity loci slowly but steadily, hopefully canola will become more resistant to the pathogen. In the legume space, there's some nice research out of the US where they've found some really strong forms of resistance in soybean that are quite interesting. So that's Mehdi's team in particular that's done that work. we're only just starting to scratch the surface in developing phenotyping assays in narrowleaf lupin, where sclerotinia is a big issue. We've done a little bit of work on sclerotinia and chickpeas, but again, it's minor QTLs that explain, 10, 13% of the variants that, don't set the world afire and obviously hard to breathe for.
Okay, I see a raised hand from Nicholas Larkan, so please unmute your mic and talk, Nicholas.
Hey, Lars. Thanks for the talk. I've got a couple questions. With your Ethiopian isolates where you failed, the PCR failed, did you get to the bottom of that? Do you see RIP mutations there or something?
So yes and no. That's literally data from a few weeks ago, and what we're waiting on is the Sanger sequencing results to come back. So what we've done is we've amplified the full-length gene and sent it off for Sanger sequencing. So you paid good attention. That data wasn't on that phylogenetic tree. That's literally something that has come through in the last few weeks. So we're hoping to get an answer. So there will be another variant most likely of the ILSW-s3e3 protein.
And I guess my other question is kind of philosophical. As a molecular geneticist myself, at what point are you comfortable start calling your major QTL at the end there and start referring to it as an actual major resistance gene rather than your best QTL?
Yeah.
If you mentalized your data and had a binary data set instead of a quantitative data set, you still probably map that pretty clearly, right?
Yeah. So
and you're hunting for a major resistance gene.
They're essentially, particularly the one in 7537, the response is so strong. It seems to be a major gene. It's indeed a nice debate to have. Usually with large effect QTLs that explain a large percentage of the variance, it's usually when we're talking 80-90% of the variance, it's usually a major resistance gene, and we can call it that. We'd like to have that evidence and clone the gene and or use EMS mutagenesis to create alterations to that version of the gene to prove that that's the causal gene, that if you don't have it, you lose that resistance. So that's where we want to get to, I guess. So I hope that answers your question. The other approach that we're taking is we're trying to see if we can purify the ILSW-1 protein, so we can infiltrate pure protein into lentil populations. And when we do that, that's a specific interaction of one protein with a corresponding receptor in the host, right? And so it's kind of Mendelizing the interaction, because there's obviously multiple genes like that in Ascochyta lentis, but that will allow us to then specifically identify one major receptor that interacts with a corresponding avirulence gene. So we're hoping to get that done in the coming years as well.
Yeah, that would be fun. That was my third question as well. Have you been thinking about any transgenic options to prove your gene hypothesis?
Yeah. So the ILSW-1 proteins, so the different versions, they are quite challenging to produce them in E. coli. In fact, we haven't been successful. So we're now trying to see if we can get that done in yeast and in trichoderma, so two different protein expression systems, and see how well that goes. So that's work in progress at the moment. So, but yeah, I guess the other approach to try and fish out the major receptor is through yeast hybrid studies, where you have your ILSW-1 protein as your bait. And you try and identify the corresponding receptor in the host that way, and that's work that Nimlash is starting in our team.
Right. Yeah. Oh, you. Sorry, I don't want to monopolize the conversation, but yeah, I mean, you're putting all your eggs in the basket of being a direct interaction, doing it that way, of course. But yeah, anyway, great talk. Thank you very much. And I'm very much hoping to see you in Perth in September.
Yeah. I missed you in Brisbane.
Yeah,
I saw you on the.
I'll get my passport sorted out this time. So.
Yeah. Well, look forward to welcoming you in Perth in September.
Yeah, cheers.
Okay. Meanwhile, I put a link for the feedback form, so please give us feedback about how things went and if everything was smooth. Let's see if there are other questions from the audience. Okay, so it seems to be done. Thanks again, Lars. It was super interesting. For those also your colleagues that missed it because of the time zone, the webinar will go up on our website and the YouTube channel in probably tomorrow. And I will let you all know via the usual channel. Thanks again.
Thank you,
Bye.
Nelson, and enjoy the rest of your afternoon.
Sure.
Yeah. Thanks. Yeah.
Bye.
Bye bye. Thank you.
Bye bye. Thank you.
Bye bye.